Hello and welcome to Chapter 30, Reproduction and Domestication of Flowering Plants in Biology in Focus. This is a lecture for John Tyler Community College. My name is Mr. Sparks and I will be guiding you through this lecture. Okay, so this is the overview. We're going to talk about how plants and flowers, plants and animals interact. Insects help angiosperms to reproduce sexually with distant members of their own species. The classic example is bees and flowers. There's one example of the longhorned bee in which the male bee is sent out to forage, unlike the classic European bee, Apis melliflora. The longhorn bee, Eucera longicornis, mistake the ophrys flowers for females and attempt to mate with them. The flower looks kind of like a female longhorn bee, and interestingly, it actually produces a pheromone or a chemical scent that deceives the bee into thinking that it is a female bee. The flower is then pollinated in the process. And unusually, the flower does not produce nectar, and the male receives no benefit. So in this relationship, it's completely the deception of the flower on the bee. The bee, all the bee does is receive the pollen sac, and then it takes it to the next flower, where it attempts to mate with that flower, and on down the road. So there's no mutual beneficially relationship there. Here is the Eucera longicornis. You can see the longhorn bee is aptly named. It has these long antennae here. And here is a pollen grain that has been attached to the head of the longhorn bee. And here's the flower Ophrys. It's a type of orchis flowered. And you can see it does have a shape that's similar to the bee, but it's also producing these pheromones that are attracting the bee into this false mating attempt. Many angiosperms lure insects with nectar or pollen. Both plant and pollinator benefit. Participation is mutually beneficial relationships with other organisms is common in the plant kingdom. Angiosperms can reproduce sexually and asexually. Angiosperms are the most important group of plants in the terrestrial ecosystems and in agriculture. So even though angiosperms are relatively new on the plant scene, evolutionarily speaking, they rapidly radiated into all kinds of ecological niches, and they have become extremely important to agriculture. So basically, all agricultural products are angiosperm products, with the exception perhaps of pine nuts. I don't know if anybody are familiar with pine nuts. You can have pine nuts with pesto, and pine nuts are very expensive, and they are a product of gymnosperms. They're a product of a certain pine tree that grows in Arizona and New Mexico. But apart from that, basically every plant product that you can think of is an angiosperm. Flowers, double fertilization, and fruits are unique features of the angiosperm life cycle. So angiosperms all share these characteristics. Plant life cycles are characterized by the alternation between a multicellular haploid generation and a multicellular diploid generation. Diploid sporophytes produce spores by meiosis. These grow into haploid gametophytes. The gametophytes produced by haploid gametes by mitosis, and fertilization of gametes produces a sporophyte. So this is just like the alternation of generations that we studied in the previous chapter, with a few distinct characteristics that we're going to talk about more in depth. In angiosperms, the sporophyte is the dominant generation, the large plant that we see. It's the tree, it's the oak tree, it's the maple tree, it's the tomato plant itself. That's the sporophyte. It's the dominant generation. The gametophytes are reduced in size and depend on the sporophyte for nutrients. The angiosperm, okay, so the gametophytes are just like the sperm inside the pollen and the ovaries and the eggs inside the ovaries. The angiosperm life cycle is characterized by 3 F's: flowers, double fertilization, and fruits. So when you see these characteristics, you know you're talking about an angiosperm, particularly flowers. Flowers are the most obvious part of the angiosperm life cycle. Flowers are the reproductive shoots of the angiosperm sporophyte. They attach to a part of the stem called the receptacle. Flowers consist of 4 floral organs: carpels, stamens, petals, and sepals. Carpels and stamens are reproductive organs, and sepals and petals are the sterile organs that just surround the reproductive organs. It's not like they don't have any function. They do have a function. Their function is to help make the carpels, stamens, carpels and stamens more attractive to pollinators in particular. Okay, here's a generic flower. It looks kind of like a lily. It might be a monocot because it's got everything is a 3-fold symmetry here, which is a characteristic of monocots. It's got 3 petals here, and there's probably 3 more petals on the other side of this flower. We've got 6 anthers, 6 filaments, and 6 anthers. These are the stamens. The filament is easy to remember because it's a filament just like a filament is a thread or a filament in a light bulb. Okay, back in the olden days, we used to have light bulbs that had little wires that ran through them, and those little wires are under tension. They are called filaments. At the tip of the filament is the anther, and this is the organ that produces the pollen. And together, the filament and the anther are called the stamen. The stigma is the tip of the female reproductive organ. This is where pollen will land, and pollen produces a pollen tube that grows down through all the way through the stigma and into the ovule, there where it will fertilize the megasporangia, and become the seed. Okay, this is the style. It's the name of the longer portion of the stigma of the female reproductive organ. And this is the ovary, which houses the ovules. The ovary is the kind of bulbous part. This is where the seeds develop. And here are the ovules. Each one of these ovules will become a seed. And you see that 3-part symmetry. It's got 6 seeds, 6 anthers, 6 petals. Probably a monocot that we're looking at here. The sepal they're like the petals. They help protect the flower when it's growing. When they are photosynthetic, and when the flower is in bloom, they just rest at the sides of the flower and help support the flower structure. A carpel has a long style with a stigma on which pollen may land. At the base of the style is an ovary containing 1 or more ovules. A single carpel or group of fused carpels is called a pistil. A stamen consists of a filament topped by an anther with pollen sacs that produce pollen. Complete flowers contain all 4 floral organs. Incomplete flowers lack 1 or more floral organs, for example, stamens or carpels. Clusters of flowers are called inflorescence. Flowers of a given plant species typically appear synchronously, promoting outbreeding. The the transition, so all the flowers of a given species, when you drive by on the roadside and you see all the hawthorn flowers are in trees are in bloom at the same time, or all the oak trees or all the maple trees, all the flowers in a garden of a particular type, they tend to all the tulips will bloom at once, all the irises will bloom at another time. They all they flowers of a given plant species typically appear synchronously, and that promotes outbreeding within that species. The transition from vegetative to reproductive growth is triggered by an environmental cue and internal signals. Floral organization is regulated by the products of floral identity genes. Mutations in these genes cause abnormal floral development. Okay, so here are PE stands for petal, SE stands for sepal, CA for carpel, and ST for stamen. So you see here's a normal Arabidopsis flower. This is a very highly studied plant. Its genome is one of the first organisms to be have its genome mapped. And this is an abnormal or mutated Arabidopsis flower. It's got, you see it has double set of petals here, and then it's got sepals and petals, but it's lacking the carpels and stamens. So it's got the carpels and stamens instead mutated and turned into a whole nother flower. So these kind of mutations do occur. The ABC hypothesis explains the formation of the 4 types of floral organs through the regulatory activity of 3 classes of organ identity genes. Each class of organ identity genes is switched on in 2 specific whorls of the floral meristem. A genes are switched on in the outer 2 whorls, the sepals and petals. B genes are switched on in the middle 2 whorls, the petals and stamens. And C genes are switched on in the inner whorls, the stamens and carpels. Now you can see here, just from this verbal description, that these genes overlap in their function. So A genes function only on sepals, but they also function on petals, and B genes function on petals as well as stamens. So A and B genes function on petals, and C and B and C genes function on stamens. So these genes cooperate on creating these certain characteristics of the flower, these parts of the flower. So individuals lacking A, B, or C gene activity will develop abnormal patterns of floral organs. So we can see in the absence of these genes, we can understand how they work more effectively together. Okay, here is a big diagram. It shows your sepals on the outside, petals on the inside, stamens next, and carpels on the inside. And here are the genes when they overlap. A genes affect the sepals. A and B affect the petals. B and C affect the carpels, and C alone affects the carpels. Okay, so I may have said one of those incorrectly, but bear with me here. Okay, so here is a map of the genes. These are all the genes that are working properly. Here's the A genes here, with the B genes overlapping, the C genes with the B genes overlapping, and then on the other side of the flower. Okay, so this is like a schematic of how the gene works, and matching it to these pictographs down here at the bottom. So if we remove the A genes, then they're replaced by the C genes altogether, and that mutant lacking an A will produce carpels all throughout where the sepals should have been. But the B and C, the anthers the stamens will be produced normally because the B and C function together. But they duplicated their site here. Okay, so lacking the B gene, we no longer have the stamens. Instead, we just have the sepals because the B and C function together to make the petals. So in the absence of B, you have no petals. You just have sepals and carpels on the inside. Then in the absence of C, you can have the absence of carpels, and you have double petals and double sepals. So you can see how these genes work together, especially if you're able to remove one gene or the other and then view the offspring. Okay. Studies of plant mutations reveal that 3 classes of organ identity genes are responsible for the spatial pattern of floral parts. These genes, designated A, B, and C, regulate expression of other genes, responsible for development of sepals, petals, stamens, and carpels. Sepals develop from the meristematic region where only A genes are active. Petals develop where both A and B genes are expressed, and stamens arise where B and C genes are active. Carpels arrive only where C genes are expressed. Okay, so notice the normal wild type plant and then compare that to the mutants. Development of the female gametophytes, or embryo sacs. An embryo sac, or female gametophyte, develops within the ovule. Within an ovule, 2 integuments surround the megasporangium. One cell in the megasporangium undergoes meiosis, producing 4 megaspores, only one of which survives. The megaspore divides, producing a large cell with 8 nuclei. This cell is partitioned into a multicellular female gametophyte, the embryo sac. Okay, so I think I want to see this whole diagram. Okay. In the megasporangium, here we're going to start here with the megasporangium. In the megasporangium of each ovule, the megasporocyte divides by meiosis, producing 4 megaspores. One survives and gives rise to the female megaphyte. So when the megasporangium is produced inside the ovule, it's producing 4 nuclei and 4 megaspores, and only one of these megaspores is going to survive to become the basis of the embryo. Okay, so let's go up here, back up to the pollen. On the anther, each microsporangium contains microsporocytes that divide by meiosis, producing microspores. And we'll move on to the microspores. A microspore develops into a pollen grain. The generative cell of the gametophyte will divide, forming 2 sperm. The tube cell will produce the pollen tube. Okay, now number 4, we're going to go back down here. Okay, so here's the pollen grains. They're fertilizing the stigma, and they grow a pollen tube, which is it's got a little tube nucleus at the tip of this. It's complicated, but it's easy to understand. I'm going to post some videos about this in the lab section on this when we talk about plant generation. That will help you to understand this a little better. But anyway, the pollen grain lands here. The pollen has 2 sperm plus a tube nucleus. The tube nucleus burrows its way down into the region where it finds the ovule, and then this is where it begins to fertilize the ovule. So after pollination, eventually two sperm cells are discharged into each ovule. Okay, right down in here. This is where the discharged, so the discharged sperm nuclei comes down into the ovule, and it fertilizes one of these synergids. Double fertilization occurs. One sperm fertilizes the egg, forming a zygote, and the other sperm fertilizes the central cell, forming the endosperm, which is, in this example, is triploid. So one of these pollen sperms fertilizes the embryo right here, and this becomes the zygote. And then another of those, the second pollen sperm cell, fertilizes the endosperm, which is this larger mass of material here, and then this becomes the endosperm. This is what grows and nourishes the seed of the plant. So when the plant, one of the advantages that the angiosperm has is that it can be dormant for a period of time, and then it will, and then when it comes time to sprout, when conditions are proper, it can feed off of that endosperm when it's beginning to grow, and it grows up through the ground and into the light, and then it starts growing its leaves. Okay, the zygote develops into an embryo that is packaged along with food into the seed. The fruit tissues surrounding the seed are not shown. When a seed germinates, the embryo develops into a mature sporophyte. So this is the mature sporophyte that comes out of the seed. So this is the diploid structure. The diploid structure then gives rise to another flower, and the flower gives rise to eggs and pollen, megasporangia and microsporangia, and the cycle continues again. Okay, this diagram is on page 600 in your textbook. If you need a little help understanding, I recommend you check that out. Okay, here's the microsporangia, the pollen, megasporangium being produced in each of the ovules. Okay, so these are different cells inside the megasporangium. These are the egg is right here in the middle. The synergids, these are just like assistant cells on either side of the embryo, and then these antipodal cells don't serve any major function, but these polar nuclei in the center of the cell, those become the foundation of the endosperm. Okay, so here's the the sperm cell pollen tube comes in, pollinates the egg nucleus. Okay, these synergids on either side are not really functional. They're just part of developing the embryo. I mean, just part of developing the megasporangium. Okay, the fertilized egg is now a zygote. The fertilized endosperm is now the nucleus of the developing endosperm. So it's like almost like its own organism here, but it produces the nutritious material that's necessary for the seed to germinate. Okay, here's the embryo of the zygote. It grows into its form. It's got this one is a dicot. It has 2 cotyledons here, and it brings into the so this is basically the seed. The outer integument becomes the seed coat. Okay, the development of male gametophytes in pollen grains. Pollen develops from a haploid microspores within the microsporangia or pollen sacs of the anthers. Remember, they're on the end of the filament. Each microspore undergoes mitosis to produce 2 cells, the generative cell and the tube cell. A pollen grain consists of the 2-celled male gametophyte and the spore wall. If pollination succeeds, a pollen grain produces a pollen tube that grows down into the ovary and discharges two sperm cells near the embryo sac. In angiosperms, pollination is the transfer of pollen from an anther to a stigma. Pollination can be by wind, water, or animals. Most angiosperms depend on insects, birds, or other animal pollinators. Okay, we're going to talk about pollination by different forms here. Here's a hazel tree. It's a staminate flower. This is the stamens only, and the carpellate flower is what receives the pollen. You can find analogous flowers like this on oak trees. They're called catkins, and these catkins are the male organ producing the pollen, and then the carpellate flowers receive the pollen. Dandelions are pollinated by bees and other insects, and dandelions they have a different appearance. Ultraviolet light is the spectrum that bees and other insects tend to see in. So when they look at a dandelion, they're looking at a more inviting pattern than we are. Not that dandelions aren't pretty, but the insects have a different perception of them. Okay. Other wind-pollinated plants include corn and wheat. Those are angiosperms that are important to agriculture. Okay, here's a long-nosed bat feeding on a cactus at night. So flowers are pollinated by birds and bats. It's interesting to note that bats pollinate the agave flower. Agave is a commercially important flower or plant that is used to grow tequila. So agave plants are dependent on bats for pollination, and that is the relationship that you might be interested in. So agave plants dependent on bats, and that's the produce of tequila. Hummingbirds drink nectar of flowers. Usually these flowers are specially adapted. Plants that attract bats are usually heavily scented to attract the bats at night. The bats like a nice fruity smell so they can come in and they rub their noses around in the anthers of the flower, and then they transfer that pollen to the next plant. And then here you can see the hummingbird drinking the nectar of the columbine flower. These are specially adapted flowers. They're at the end of the reservoir is there will be some nectar, and then the hummingbird will dip its beak into the flower and push its way all the way up into the flower, and then it will get pollen from the anthers onto its face, and then it will go visit another flower and it'll take that pollen to a whole other flower. It's the same basic phenomena, the way that insect pollination works. Okay, so here's the long-nosed bat feeding on a cactus flower. Abiotic pollination by wind occurs in angiosperms, including grasses and many trees. Wind-pollinated angiosperms produce small, inconspicuous flowers that lack nectar or scent and release large amounts of pollen. In fact, you probably didn't even notice this, but grasses do have flowers, and those corn is a grass, and the silk on corn is the stigma of the corn. When the male corn flower disperses its pollen in a cornfield, it gets picked up by that silk, and that silk transfers it to the seed, where the seed then becomes fertile. Pollination by insects includes bees, moths, butterflies, flies, and beetles, and occurs in about 65% of all angiosperms. Bees are the most important pollinators, especially for human agriculture. Bees are critical to human agriculture. Basically, all of the fruits and vegetables that we're familiar with are pollinated by bees. Floral adaptations to attract bees include production of nectar, sweet fragrance, brightly colored flower petals, and nectar guides. Nectar guides are like pathways into the flower. Pollination by bats occurs in plants that produce light-colored aromatic flowers. Pollination by birds occurs in plants that produce large, bright red or yellow flowers with a little odor and large quantities of nectar. The petals of bird-pollinated flowers are often fused into a floral tube. In fact, it's an easy and fun thing that you can do is feed hummingbirds later on in the spring when the temperature gets nice. The hummingbirds are migrating back from the southern United States and from Central America, and they're really hungry when they get here. And you can mix up sugar water. You want to mix a lot of sugar into some water, and then you put it in. You can buy a commercial hummingbird feeder at Lowe's or Home Depot, and you can mix up this sugar water, and it will attract hummingbirds right to your porch. And once they get used to it, they're almost fearless. They'll come and obtain that nectar, that sugar water, without any problem at all. It's really, really fun thing to watch. Okay, double fertilization. After landing on a receptive stigma, a pollen grain produces a pollen tube that extends between the cells of the style and the ovary. Double fertilization results from the discharge of two sperm from one pollen tube into the embryo sac. One sperm fertilizes the egg, and the other sperm combines with the polar nuclei, giving rise to the triploid food-storing endosperm. Okay, the endosperm is the meaty part of the seed. When you're eating beans or corn, you're eating the endosperm, primarily the endosperm of the plant. Okay, so here's the pollen grain landing on top of the stigma. It builds the pollen tube. It excavates its way. There's three nuclei, the two sperm nuclei and the tube nucleus, and travels down through the style. It travels around the ovary and down into the micropyle. This is the opening, and then into the egg. Okay, and the egg here, it's got the synergid on either side, and it's got these antipodal cells. These are kind of cells that are not, they lose their structural function after fertilization. They become, they just basically go by the wayside. And then here's the polar nuclei. These are the two nuclei inside. This becomes the endosperm when it's fertilized by the second pollen sperm. First pollen sperm fertilizes the egg. Second pollen sperm fertilizes the polar nuclei, and then this becomes the triploid endosperm. Okay, here's a better description. All right, here we got a pollen tube comes down, enters through the micropyle into the egg. There's the synergid on either side, the polar nuclei, the second sperm nucleus, these two polar nuclei plus the sperm, and this generates the endosperm. That's the bean part of the bean right here. This is the bean part of the bean right here in the middle of the seed here. This is the integument. This becomes the outside, the hard coating of the seed. Okay, so here's the zygote. This is the diploid zygote, which eventually becomes the sporophyte. So that's just going to become your sporophyte right there. And the endosperm is meaty and nutritious, but it gets used up by the zygote right away after it fertilizes and after the seed starts to grow, and then it gets used up by that zygote, and then it just goes by the wayside. After double fertilization, each ovule develops into a seed. The ovary develops into a fruit enclosing the seed. Endosperm development usually precedes embryo development. In most monocots and some eudicots, endosperm stores nutrients that can be used by the seedling. In other eudicots, the food reserves of the endosperm are exported to the cotyledons. The first mitotic division of the zygote splits the fertilized egg into a basal cell and a terminal cell. The basal cell produces the multicellular suspensor, which anchors the embryo to the parent plant. The terminal cell gives rise to most of the embryo. The cotyledons form and the embryo elongates. By the time the ovule becomes a mature seed and the integuments harden and thicken into a seed coat, the zygote has given rise to an embryonic plant with rudimentary organs. Okay, so here's the fertilized. These are the stages that it goes through: the fertilized zygote, terminal cell, and a basal cell. The terminal cell begins to develop into the proto-embryo. And the pro-embryo becomes the cotyledons, and here is the generic seed here, and it's got the cotyledons here. The shoot apex is right in the middle. That's the apical meristem right here in the middle, and the root apex is down here at the bottom. This is all inside the seed coat, inside the integument, surrounded by the endosperm. And this is the suspensor, the part of the embryo that attaches it to the plant until the seed dries up and breaks off and finds its way to a fertile location. The embryo and the food supply coat are enclosed in a hard protective seed coat. The seed enters a state of dormancy wherein it stops growing and slows metabolism. A mature seed is only about 5 to 15% water. A mature seed generally dries out quite a bit, and in being dried out, it actually prepares itself for conditions that when there's enough moisture around it, that seed will draw the moisture into it because it's so dry, and then it will begin to sprout. In some eudicots, such as the common garden bean, the embryo consists of the embryonic axis attached with two thick cotyledons. Below the cotyledons and the embryonic axis is called the hypocotyl and terminates in the radicle, the embryonic root. Above the cotyledons is called the epicotyl. The plumule comprises the epicotyl, young leaves, and shoot apical meristem. Okay, so here's some graphics describing this in more detail. The common garden bean is a eudicot with thick cotyledons. The fleshy cotyledons store food absorbed from the endosperm before the seed germinates. So here's the epicotyl. This is where the little leaves are going to sprout out, and here's these are cotyledons. They are functional. They're functional. They will become functional leaves, the first leaves. But this is where most of the nutrition of the endosperm has been absorbed into the cotyledons. The fleshy cotyledons store food absorbed from the endosperm before the seed germinates. This is the castor bean down here. It's a eudicot with thin cotyledons. The narrow membranous cotyledons shown in the flat views absorb food from the endosperm when the seed germinates. So in this case, the endosperm is more prominent. The endosperm is in blue here. And then this is the endosperm of a corn. Like all monocots, corn has only one cotyledon. Maize and other corn and other grasses have a large cotyledon called a scutellum. The rudimentary shoot is sheathed in a structure called the coleoptile, and the coleorhiza covers the young root. Okay, so here's the endosperm of the corn. That's like the nutritious part of the corn here. You don't want to eat castor beans; they're very poisonous. In the common garden bean, this is the endosperm was in this area, but now it's been absorbed by the cotyledon. So you're absorbing the energy of the endosperms as it's been transferred to the cotyledons. The seeds of some eudicots, such as castor beans, have thin cotyledons. A monocot embryo has one cotyledon. Grasses, such as corn and wheat, have a special cotyledon called a scutellum. Two sheaths enclose the embryo of the grass seed. A coleoptile covering the young shoot and a coleorhiza covering the young root. Seed dormancy increases the chance. Okay, so seeds, the basic advantage of a seed is that it can wait for a long time for conditions to be proper before it sprouts, before it tries to grow into a new tree. With non-vascular seedless plants and vascular seedless plants, the bryophytes and the ferns, those spores have to find places to grow immediately after they are produced. But with seeds, they can just they can wait until the conditions are proper for the right time and place. So seed dormancy increases the chances that germination will occur at a time and place most advantageous to the seedling. The breaking of seed dormancy often requires environmental cues such as temperature or lighting change. So seeds won't sprout until the spring when the conditions are right. That they have to have the right amount of rainfall and all different kinds of conditions. In fact, what's interesting is some seeds won't even sprout unless they're treated in certain ways. Like some seeds you have to rough up on sandpaper, and some seeds have to be soaked like 3 times before they'll sprout, and some seeds have to be swallowed by an animal, like the mango. Mangoes have to pass through a cow in order for the tree to be able to, sprout. The breaking of seed dormancy often requires environmental cues such as temperature and lighting changes. Germination depends on imbibation, the uptake of water due to low water potential of the dry seed. The radicle, the embryonic root, is what emerges first. Next, the shoot tip breaks through the soil surface. In many eudicots, a hook forms in the hypocotyl, and this growth pushes the hook above the ground. Light causes the hook to straighten up and pull the cotyledons and shoot tip up. Okay, so this is like especially in dicots, like in our bean here. These are the cotyledons. These first. So what happens first is the radicle, the tip of the root comes out, and that's the root apical meristem begins growing the root into and branching into these different forms. And here it's probably forming its first mycorrhizal relationships and beginning to transfer water and nutrients into the plant. And then the cotyledon is the first leaves that come out of the the plant. They begin to photosynthesize. They turn green, and then these cotyledons will shrivel up and drop off and give rise to regular foliage leaves. These foliage leaves are the leaves that we're all familiar with. So if you take a look at a seedling as it's sprouting out of the ground, especially a dicot, you will find these cotyledon leaves. Okay, so the hypocotyl, the radicle. The radicle is where it starts growing out. The hypocotyl is kind of like the bend in the plant, and then it pushes up and then it pulls out the cotyledons, and then it grows. The hypocotyl it becomes the main stem of the plant. Oh, it's the same thing for the corn. Okay, except for corn, instead of pulling the hypocotyl out of the soil, it'll push the coleoptile out of the ground, and then that forms the basic foliage leaves form kind of right away. In some monocots, such as corn and other grasses, coleoptile pushes up through the soil. Fruit form and function. A fruit develops from the ovary. It protects the enclosed seeds and aids in seed dispersal by wind or animals. A fruit may be classified as dry if the ovary dries out at maturity, or fleshy if the ovary becomes thick and soft and sweet at maturity. Fruits are also classified by their developmental origins. They can be simple from a single or fused carpels, aggregate from a single flower with multiple separate carpels, or multiple from a group of flowers called an inflorescence. Okay, here are some different fruits. A pea fruit is a simple fruit. A simple fruit develops with a single carpel or several fused carpels and one flower, like a pea or citrus or a peanut. The aggregate fruit develops from many separate carpels of one flower. Examples are raspberry, blackberries, and strawberries. A multiple fruit develops from many carpels of the many flowers that form an inflorescence. An example is the pineapple. And an accessory fruit develops largely from tissues of other than the ovary, and the apple. And in the apple fruit, the ovary is embedded in a fleshy receptacle. Okay, so most of the all right. Here's the ovary of the apple, and then surrounding that, this area surrounding here is what becomes the fruit. An accessory fruit contains other floral parts in addition to ovaries. Fruit dispersal mechanisms include water, wind, and animals. Basically, any way that fruit can get around. Dispersal by water, dispersal by wind. Okay. Okay, so like a coconut seed is an embryo that travels around, and if you've ever been to the Caribbean, you'll see coconut palms on the beaches. They're very common. These coconuts fall into the water and they float, and then they get washed around by the ocean, and they wind up on a distant shore somewhere far away before they find proper conditions to grow. Many plants are dispersed by wind. Maple trees are a classic example around here. Dandelion seeds, which are actually one-seeded fruits. Each one of those seeds is a fruit. Tumbleweeds, they blow all around the southwest, and they carry seeds with them, and those seeds get dispersed as they travel. Dispersal by water occurs in buoyant seeds like fruits like a coconut, which can survive for long periods at sea. Okay, so there's the coconut seed embryo. The endosperm of the coconut is the white part of the coconut when you crack it open and eat it. I don't know who's eaten a coconut, but they can be delicious. It's easier to buy the shredded coconut, but you can buy a coconut and crack it open with a hammer, and you dig out the endosperm, and it's delicious that way. Dispersal by wind occurs in seeds and fruits that have adaptations such as parachute or wing-like structures. Okay, here's a giant seed of a tropical Asian climbing gourd. Probably never going to see this in your lifetime, but you will see other plants that are dispersed by wind. Okay, dandelion seeds, the winged fruits of a maple. These are all much more common things that you're going to see. The tumbleweed, like in the Southwest, they blow through. If you ever watch an old Western, they always have a tumbleweed blowing down the middle of the street. Dispersal by animals occurs in seeds and fruits that are edible or adapted to attach to an animal's skin or fur. So a lot of these fruits, like choke cherries and a lot of the wild berries that grow around in the area, they are eaten by birds or eaten by mammals, and like raccoons or bears, and then their seeds are dispersed by defecation. So that bird will fly away and that defecate that seed, and that seed will find purchase in a particular location that's favorable to its growth, and then it will grow into a new plant. Okay, so here are. I think this puncture vine is pretty impressive. I've never seen one in real life, but it's obviously it's a burr. It gets into your clothes. It gets into your fur. I don't know how furry you guys are, but if you're an animal, you have fur, and you get this is basically like a burr. So you're probably familiar with burrs. There are many different kinds in our region, and they get on your clothes, and then you pick off those clothes, and then by the time you move to a different location, that means that plant that seed has found a new location to try to grow. Squirrels hoarding seeds or fruits are pretty common. This is just like squirrels hoard acorns. They'll find acorns in the fall when the acorns fall off the trees. They'll eat a bunch of them, and then they'll take some of them and try to bury them to get through the winter, and they'll bury them in certain locations, and that can actually help the acorn itself to sprout. So the squirrel is kind of working on behalf of the oak tree. Here's seeds dispersed in black bear feces. I did a study on coyotes in West Virginia one time, and part of that study was collecting coyote feces, and we did find a lot of chokecherry and a lot of other native berries in the feces of the coyotes that were in the area. If you do wildlife work, a lot of it is studying feces, believe it or not. And in fact, when I worked in Brazil, we collected a lot of feces from the jaguar and the ocelot, and we analyzed those feces and tried to find out what animals they were eating. So they were eating wild pigs, peccaries, agoutis, all different kinds of small mammals that lived in that area. Okay, here's an ant carrying a seed with an attached food body. So the idea here is that a seed will have like a little bit of fruit nutrition attached to it, and then it will entice the animal like an ant to pull that seed down into its burrow, and then it will eat that food body, and it'll leave the seed in a condition where it's appropriate for it to sprout. Fruit of the puncture vine looks pretty terrifying to me. Would be bad if you stepped on this. Let's put it that way. Here's a squirrel hoarding seeds or fruits underground. Okay, this is a red squirrel, and it's got a lot of pine cones around it, so it's probably eating pine nuts. There's the black bear feces. There's the ant carrying with food with a seed with an attached food body. Flowering plants reproduce sexually, asexually, or both. Many angiosperm species reproduce both sexually and asexually. Sexual reproduction results in offspring that are genetically different from their parents. Asexual reproduction results in a clone of identical organisms. So these are two different ways. The advantage of sexual reproduction is you get genetic diversity, and that can adapt to all kinds of different conditions that are unforeseen in the future. And asexual reproduction is basically cloning or creating identical individuals, which is advantage if conditions are stable, but it's not an advantage if conditions change. For example, one disadvantage of cloning species or asexual reproduction is if there's a particular disease that comes along that can destroy your species. If you don't have genetic diversity built into your population, then you're going to be susceptible to that disease. Your whole population, because it's the same, might be susceptible to the disease in the same way. So asexual reproduction does occur, though. Fragmentation is the separation of a parent plant into parts that develop into whole plants. It's a very common form of asexual reproduction. In some species, the parent plant's root system gives rise to adventitious roots that become separate shoot systems. Okay, fragmentation is when you can break off a part of a plant and grow it. It's the basic premise behind grafting. I think we're going to talk about grafting later on in this chapter. But these are two mechanisms of asexual reproduction. Okay, so here's asexual reproduction in aspen trees. Aspen trees are well known to reproduce through adventitious roots. The aspen tree will shoot roots. It'll grow a whole new tree off of the end of its roots. So all these aspen trees here are really only one tree. And we have a plant here in Virginia that grows like this called the pawpaw. It typically grows along it grows in wooded areas and along the James River. If you can find pawpaw trees at the right time in the fall, they have delicious fruits that are they taste like mangoes. They're called custard apples by the local people in the Appalachian Mountains. And they are clonal species as well that grow by adventitious roots. So basically, all these aspen trees here are all clones. This is all one tree. Even though this looks like this is one tree here, its adventitious roots grow up and give rise to all these other aspen trees here. Same way with the pawpaw. If you're interested in more about the pawpaw, I can tell you about it in class. Apomixis is the asexual reproduction of seeds from a diploid cell. So apomixis is when it's basically selfing. It's cloning the seeds, but it uses the seed and dispersal mechanism as a way of reproduction. Advantages and disadvantages of asexual versus sexual reproduction. Asexual reproduction is called vegetative reproduction. Asexual reproduction can be beneficial to a successful plant in a stable environment. However, a clone of a plant is vulnerable to local extinction if there is an environmental change or a a disease or any challenges to that environment. Sexual reproduction generates genetic variation that makes evolutionary adaptation possible. However, only a fraction of seedlings survive. Some flowers can self-fertilize to ensure that every ovule will develop into a seed. Many species have evolved mechanisms to prevent selfing. Okay, so the way plants do this is they'll typically they can flower at different times, or they can the flowers on they have plants with one type of flower, male flowers on one plant and female flowers on the other plant, or they can have the structure of the flower can be such that it prevents that selfing. I think we're going to go into that in a little more detail here. Mechanisms that prevent self-fertilization. In angiosperms, many angiosperms have mechanisms that make it difficult or impossible for a flower to self-fertilize. Dioecious species have staminate and carpellate flowers on separate plants. So the male and female flowers will be on different plants. This is particularly common in cedars, red cedar and Himalayan cedar. They're two trees that they have male flowers on one plant and female flowers on another plant, and that helps to keep them separate genetically. That is called dioecious. Staminate flowers on one and carpellate flowers on a dioecious plant. Okay, so this is an arum. Sagittaria latifolia, the common arrowhead. It's dioecious, so it has plants that produce only staminate flowers on the left and plants that produce carpellate flowers on the right. And some species, such as the alpine wood sorrel, produce two types of species on different individuals called thrums, which have two types of flowers on different individuals, which have short styles and long stamens, and pins which have long styles and short stamens. The insect foraging for nectar would collect pollen on different parts of its body. Thrum pollen would be deposited on pin stigmas and vice versa. So here you've got your stamens here. You have your little insect crawling in here. It gets the pollen on it in this way. But the styles are located way deep down in the flower. And then in another flower, on another plant, there's another flower where the styles reach up, and the pollen from this plant will get onto these styles, but it has to reach down far to get the stamens here, and then it gets pollen from the stamens here, and then when it reaches down to the other plant, it fertilizes those styles here with the pollen from this plant. Okay, so plants have different strategies to avoid self-pollination. Others have stamens and carpels that mature at different times or are arranged to prevent selfing. The most common is self-incompatibility, a plant's ability to reject its own pollen. Researchers are still unraveling the molecular mechanisms involved in self-incompatibility. Some plants reject pollen that has an S gene matching an allele in the stigma cells. Recognition of self-pollen triggers a signal transduction pathway leading to a block in the growth of a pollen tube. So plants have molecular ways of telling what's their own pollen and what's their own pollen from their same flower or their same plant, and they're able to block it. They block the growth in the pollen tube. Totipotent cells, vegetative reproduction, and tissue culture. Totipotent cells are basically plant stem cells. They are able to asexually generate a clone of the original organism through cell division. When vegetative reproduction is induced by humans, it is called vegetative propagation. Okay, propagation is what we do to plants. We propagate plants. We cause them to grow, usually for our benefit. Many kinds of plants are asexually reproduced from plant fragments called cuttings. A callus is a mass of dividing undifferentiated cells that forms where a stem is cut and produces adventitious roots. So you can make a cutting off of a plant, and if you treat it in water, particularly with certain hormones, plant hormones can help to cause that stem to produce adventitious roots. A twig or bud can be grafted onto a plant of closely related species or variety. The stalk provides the root system, and the scion is grafted onto the stalk. So this is really common with fruit species, species that we grow for our benefit for fruits, like apples. They do this a lot with apples. You'll take an apple that has a really hardy, a hardy root system that's well adapted to your climate. Like maybe you live in the mountains and you need a root stock that's adapted to that's hardy to cold in the mountains. But you want to grow a better apple. You want to grow a Macintosh or a Honeycrisp. So you take that and you graft the fruit apple scion onto the stalk of the root system, and that enables you to grow the good fruit with the good root stock. Because the apple that you're trying to grow, let's say it's the Honeycrisp, that's my favorite. It may be better adapted to a warmer climate, but you can grow it in a colder climate by using a stock that's adapted to those conditions. So you want to have a hardy stock and a good fruit. So sometimes you don't get those characteristics in the same plant. So in order to work your way around that, you can graft that plant onto the stock. Okay, test tube cloning and related techniques. Plant biologists have adopted in vitro methods to clone plants for research or horticulture. Small fragments of a parent plant are cultured in artificial medium. A callus of undifferentiated cells can sprout shoots and roots in response to plant hormones. This is pretty easy to do with plants. You can't really do it with animals so well. Salamanders will grow new limbs. They do have undifferentiated cells. These undifferentiated cells are basically the same thing as stem cells, and they can grow these new sprouts. It's particularly if you treat them with certain types of plant hormones. Okay, so here's a cloning of a garlic plant. It just shows a mass of undifferentiated cells. I don't know where they might have taken this from the bulb of a garlic, or they grew it in a test tube somewhere, and then they begin to treat it with certain hormones. Those hormones elicit the growth of the vegetative structures from the plant, and then they cause a root to eventually to grow. So you can really play around with plants in the laboratory. Plant tissue cultures facilitate genetic engineering and the elimination of viruses. People modify crops through breeding and genetic engineering. People have intervened in the reproduction and genetic makeup of plants for thousands of years. Probably about 6,000 years. That's the oldest agricultural records of particularly wheat and lentils that were grown in the Fertile Crescent, the area that is now Turkey in the Mediterranean region. Hybridization is common in nature and has been used by breeders to introduce new genes. So it's common to take different strains of plants and within the same genus and crossbreed them and produce better strains of plants that are better able to survive. Corn is a product of artificial selection. It is unable to persist in nature. Okay, so here's the ancient corn species. This is maize. This is All corn is from Mesoamerica, from Central America. It was originally grown by the Mayans and the Aztecs, and it was taken back across the sea by Cortez and the other conquistadors as they came into contact with the native people. And then through years and years of selective breeding, the ancient Mesoamericans, the Aztecs, they built better and better. They selected for better and better traits of the corn until nowadays we have modern day corn here. And here's the silk that transfers the pollen down, and there's a silk strand for each kernel of corn, and those silk strands travel down into the pollen, and that's where the pollen tube travels down through into the corn kernel. Delicious corn, one of my favorite things. Not so delicious ancestral corn. It was probably useful on some level, but we've definitely improved it through hybridization. Plant breeding. Mutations can arise spontaneously or can be induced by breeders. Plants with beneficial mutations are used in breeding experiments. Desirable traits can be introduced by hybridizing wild species with domestic varieties within the same genus or species. Plant biotechnology has two meanings. In a general sense, it refers to innovations in the use of plants to make useful products. In a specific sense, it refers to the use of genetically modified organisms in agriculture and industry. Modern plant biotechnology is not limited to transfer genes between closely related species or genera. In traditional agriculture, in order to transfer traits between species, between plants, they have to be generally fairly closely related, in the same genus generally, or in the same family at the most. But they've got to be fairly closely related in order to transfer these traits from one with using traditional agricultural practices. Transgenic organisms are genetically modified to express a gene from another species. So, for example, they have a new tomato breed that's called the Flavor Saver tomato, and it's been bred to be resistant to cold. And you can refrigerate this tomato and travel it, and it will travel in relatively decent shape into its destination. But the way that they did that is they spliced a Arctic flounder gene into the tomato. So the tomato obviously is not very closely related to an Arctic flounder, which is a fish. So you take that with modern transgenic techniques. You can take that trait and transfer it directly. Now, me personally, I just wait until summer to eat tomatoes because the tomatoes in the summer are delicious. And these tomatoes that they ship all the way across the world, they're not. In my opinion, they're not great tomatoes in any way. Okay, genetically modified plants may increase the quality and quantity of food worldwide. Transgenic crops have been developed that produce proteins to defend them against insect pests. They tolerate herbicides and they resist specific diseases. Okay, so what you can do with transgenic plants is you can take they have transgenic corn called BT corn. Bacillus thuringiensis is a prokaryote. It's a bacterium that they have bred into the corn plant. They've taken the gene of the Bacillus thuringiensis that produces a particular type of toxin that is poisonous to the cutworm, a type of caterpillar that affects the roots of the corn plant. And it's basically the corn plant produces its own pesticide, which is great because it reduces the amount of pests that are attacking the corn plant. But as you know from your studies of evolution, those cutworms eventually develop the populations develop resistance to that Bacillus thuringiensis. So now you've got these corn that can produce their own pesticides, but then the pests are evolving to be able to outcompete that. You can also develop crops that are resistant to herbicides, like Roundup Ready soybeans. So these are soybeans that have genes in them that enable them to resist a particular particular herbicide known as Roundup. You spray Roundup over that crop, those soybean crops, and it kills all the other plants in that area. So it leaves you just with a nice, clean crop of soybeans. But the thing of that is that the plants are developing resistance to those herbicides, just in the same way that the cutworm is developing resistance to the Bacillus thuringiensis in the corn plant. So now you're getting plant weeds that are becoming super weeds, and they're able to resist that Roundup herbicide, and then no other herbicides will kill them either. So you've got like you get like a terrible problem with these Roundup with this situation where you've developed the plant. So anyway, through evolutionary forces, even though you get a couple of generations of really great agricultural product, you ultimately wind up with the same problems that you have with these plants and these animals being resistant to pesticides and herbicides in the first place. And some transgenic crops can resist certain diseases. So nutritional quality of plants is being improved. Golden rice is an example that's typically used. It's a transgenic variety of rice that's being developed to address vitamin A deficiency in the world's poor. It takes a gene from golden rice takes a gene from what is it? It's like marigolds or something like a. It's like a. Oh. Well, it takes a. Well, I'm not finding it right here, but it takes a gene from another plant, and it enables that rice to develop vitamin A richness, and it becomes actually more yellow in color. Transgenic cassava has enriched levels of nutrients and reduced levels of cyanide-producing chemicals. Cassava is an interesting plant because people have been eating cassava for thousands of years, and it's a plant that produces its own poison. Cassava root has cyanide in it, and you have to produce it. You have to prepare it in a certain way. You have to soak it in water, and you have to boil it and treat it in a certain way. Otherwise, it's poisonous. But it's one of those things, like, you have to wonder, how did people ever think of eating cassava when it's poisonous? You can't just reach in the ground and grab it and eat it like you could a potato. But it is a root plant like a potato. But it has to be treated in a certain way. Anyway, these transgenic cassavas have enhanced levels of nutrients and reduced levels of cyanide-producing chemicals. So, transgenic technologies, they do produce useful products for humanity. Okay, here's the transgenic cassava. Here's a woman in looks like South America who's harvested a lot of cassava root here, and now she's going to have to prepare it in a certain way so that it's edible. Biofuels, reducing biofuel dependency. Biofuels are derived from living biomass that the total mass of organic matter in a group of organisms. Biofuels will reduce the net emission of CO2 and greenhouse gas. Biofuels can be produced through the fermentation and distillation of plant materials such as cellulose from rapidly growing crops, including switchgrass and poplar. Okay, so these are some solutions that we have based on the plants that we're able to grow. Some biologists are concerned about releasing genetically modified organisms into the environment. What happens if they crossbreed with closely related species and impart those disease and herbicide-resistant qualities into those plants? Will those plants become superweeds? The concern originates from the unstoppable nature of the experiment. So when you're putting these genetically modified crops into the environment, and you really don't have any control because insects and the wind is blowing these different the pollen from the different transgenic plants into the neighboring fields, and they may be bringing those traits into other plant populations. One concern is that genetic engineering may transfer allergens from a gene source to a plant used for food. Okay, so you could you might be like if you have a transgenic peanut and somebody's allergic to peanuts and you transfer that gene accidentally into soybeans, well then maybe the soybean population becomes contaminated with that source. As an interesting aside, there's a fungal effect that's occurring in Florida called citrus greening, and they're able to address it with a transgenic citrus that they're developing, and the transgenic citrus has a part of a pig gene involved in it. So the question is, if you're trying to keep kosher or halal, where you are religiously not eating pork products, can you drink orange juice that has the pork gene in it, that has the pig gene in it? So it's an interesting consideration to think about. Some GMOs have health benefits. For example, the corn that produces the Bt toxin has 90% less of a cancer-causing toxin than non-Bt corn. Bt corn has less insect damage and lower infection by fusarium fungus and produces the cancer-causing toxin. Okay, so the fungus produces the toxin, and the Bt corn has lower infection rates by this fungus, and so it produces less of this cancer-causing toxin. GMO opponents advocate for clear labeling of GMO foods. So the argument here is like, okay, GMOs, let's say that they're wonderful and that everybody agrees how awesome they are, but why shouldn't we put a label on it? We should know if we're eating GMO foods or not eating GMO foods. It should be up to the consumer to interpret that information. What do you think about that? Possible effects on non-target organisms. Many ecologists are concerned that the growing of GM crops might have unforeseen effects in non-target organisms. Addressing the problem of transgene escape. Perhaps the most serious concern is the possibility of introducing genes escaping into the related weeds through crop-to-weed fertilization, hybridization. This could result in superweeds that would be resistant to many herbicides. In fact, these superweeds already do exist, and with a little bit of research, you can find out about these. I'll try to put some links on the video study guide about this topic. Efforts are underway to prevent this by introducing male sterility, apomixis, that's self-fertilization and production of seeds, transgenes into chloroplast DNA, not transferred by pollen, and strict self-pollination. Okay, let's see. What is this? This is a table of... Okay, do monkey flower species differ in allocating energy to sexual versus asexual reproduction? Okay, so here are the different monkey flower species. Here's the amount of nectar that they produce. Well, I haven't really done this experiment, so if you're interested in this, go on page 610, and it's going to show you that the production of plant weight is inversely related to the nectar volume. Okay, so you get more seeds the more nectar that you produce, but you get less plant volume. More seeds, less plants. The more nectar, the more seeds, the less plants. So greater reproductive success, the more nectar you produce, but less biomass formed. Here's your monkey flower eating hummingbird. Okay, here's the pollen tube, comes up, fertilizes the zygote. There's the endosperm nucleus, the triploid, two polar nuclei plus the sperm. That becomes the endosperm. All right, and the endosperm is produced, and then it's absorbed in certain dicots, like beans. The endosperm is absorbed by the hypocotyledons, and then those hypocotyledons produce the first leaves of the plant as they're pushed up out of the soil by the radicle.